For this Monday evening, the Cardiac Bioelectricity Seminar, we have a guest from not too far away, from Chicago, which is really close. And Bers, I don't think we need an introduction. If I start going over all his credentials, it'll take me longer than his duration of his visit here. So I think we'll leave it at that. But Don currently, he got his Ph.D. from UCLA, if I'm not mistaken. Further here, you mentioned to me one year in Colorado. Undergraduate. So, and then we actually met, I think, for the first time on CBAC, on cardiovascular studies section reviewing grants. That's very good. Maybe at happier times because the funding path was a little better than what it is now. And Don literally wrote the book on EC coupling in cardiac cells and calcium cycling, and he's been recently very interested in what goes wrong in calcium cycling during heart failure and the implications to arrhythmogenesis, as well as regulation of calcium cycling in terms of Chemokinase and other things that affect SR uptake, release, and the whole calcium cycling. So I think that's what he's going to tell us about. So please thank you for that.
Thanks very much. It's a pleasure to be here in this palace. It's a wonderful place.
The palace is because of Frank Yin, that's sitting right there. That's his.
So I decided to talk about a slice of stuff that kind of fits together, and as I'll come to, it does focus a bit on chemokinase and regulation of the heart. It also talks about heart failure. So I'll take you through my quick trip around the myocytes. So with each action potential, we activate the sodium channels that bring sodium into the cell. And that sodium entry causes the upstart of the action potential, and that depolarization, of course, activates the L-type calcium channels, which bring calcium in, and a little comes in maybe by the sodium-calcium exchange, and that activates the calcium release from the sarcoplasmic reticulum, which then goes to largely bind to the myofilaments and starts to activate the contraction. And as contraction proceeds, you reach a certain point, calcium gets taken back up by the SR, allowing the myofilaments to relax. And indeed, during this relaxation process, not all the calcium goes back to the SR. Some is transported out of the cell by this electrogenic 3-to-1 sodium-calcium exchanger. And of course, eventually all the calcium gets back off the myofilaments that went onto it at least, and then you're left with this bolus of sodium that came in during the cardiac cycle, and of course you have to extrude that to get back to the initial starting point, and that goes out almost exclusively by the sodium pump. And so you've completed that cycle where the electrical, the calcium, and the contraction is going on. And for those quantitatively oriented, each one of those balls is between 1 and 2 micromolar of sodium or calcium. So today I'm going to talk about some of the interplays in this system, and I'm going to talk mainly about SR calcium handling and how it's altered in heart failure, with a particular focus on calmodulin and chemokinase and how they modulate the ryanodine receptor. Of course, we know chemokinase also phosphorylates phospholaminin and can regulate pumping. It also participates in regulation of the L-type channel and some other channels as well. But I'm going to focus a bit on this process here. As well, at the end, I'm going to talk about chemokinase that, in another role, on another release channel, the Ip3 receptor, and how that may be involved in calcium-dependent nuclear signaling. So I'll also use this heart failure model that was developed here at WashU by Steve Pogwiz, and it's an aortic insufficiency, aortic stenosis model in rabbits. It develops over 6 to 9 months, and Steve has characterized this extensively in mapping studies, and we've worked on it together to try and understand better some of the cellular bases for what's going wrong. So these hearts in cross-section here you can see are much bigger. On the average, they're almost twice as big by the stage we study them. The cell size is also nearly twice as big, and the left ventricular and diastolic volume is bigger. They show on echo 38% reduction in fractional shortening and about 10% die of sudden death. And Steve has also measured with 3D mapping studies that 90% of these guys at the time we study them have these runs of non-sustained VT, and they're initiated by non-reentrant mechanisms, whereas we don't see any of that in the culture monitoring events studied in controlled animals. So a lot of the work that I'll talk about today is indeed at the cellular level, and we see cellular manifestations of these initiating triggered activities. In the case here, before ISO, you see contraction and calcium transients. And after stimulation with ISO, of course, we see stronger contractions and calcium transients, and also the appearance of these after contractions that are driven, here are the contractions, driven by calcium. And of course, that calcium transient here also drives the transient inward current and delayed after depolarizations. That can be one of the forms of triggered activity that occur. So we've also studied the sort of fundamental aspects of the calcium handling, and I'll go over only a couple of points on that very quickly just to set the stage. The twitch calcium transients are down in amplitude in the heart failure model. The caffeine-induced contractures or calcium transients that are indicative of SR calcium content are also reduced, both by about 40%, and so is shortening reduced at the myocyte level, and matches up approximately with that 30 to 40% reduction in shortening based on echo. So all three of these parameters are down. So we can also take as a measure of the fractional release of calcium during a twitch from the sarcoplasmic reticulum, how many little red balls were released out of how many were there, twitch over caffeine. And in fact, that kind of poor man's index of EC coupling or fractional release is unchanged in heart failure. So it doesn't look like it's a fundamental defect in the EC coupling mechanism per se, just less calcium available for release. And that may be sufficient, as you can see here, to describe the decrease in calcium transient and contraction. Moreover, the other aspect, the triggering calcium current that triggers release, is in fact almost identical in terms of the current density between the control and heart failure. And that's remarkable both for the fact that it's so unchanged every time we do this experiment, despite the fact that cells are twice as big. So kind of an interesting side point is, how do the cells know to make exactly this same density of calcium current as the cell's hypertrophy? But so that's not the answer. It's not the trigger change. It seems to be the load. And so the reasons that the load are down are probably threefold, and the balance of these contributors is probably different in different heart failure models or at different stages of heart failure. But I think they're probably generally true. That is that there's an increase in the sodium-calcium exchange expression or function, and that competes then with the SR pump for pumping calcium back into the SR. And if there's also a reduction in SR calcium pump expression or function, that also weakens the competition, so less calcium is pumped back into the SR and more is taken out of the cell. In addition, there may be an increase in SR calcium leak that I'll talk more about here, which will allow calcium to leak out of the SR, and again, all lowering the SR content, and that lower SR content will lead to systolic dysfunction, just less calcium available to release. And remember, then the fractional release was in fact unchanged. So the exchanger is interesting because it also, as well as being upregulated and competing with the SR pump, it also is the current that we believe underlies the transient inward current, that when spontaneous calcium release occurs, it drives an inward current, 3 sodiums to 1 calcium, and that produces the ITI, which drives the delayed after depolarization, and if big enough, could trigger an actual potential here. So that increases the propensity for triggered arrhythmias. And you couple that with the nearly 50% reduction in the IK1 current that stabilizes the resting membrane potential and holds it near EK. That means that any given amount of ITI will cause a greater depolarization and again increase the likelihood for a DAD to trigger an action potential. So these are both contributing to the increase in the propensity, but they wouldn't happen, these DADs, unless you get release of calcium from the SR. And one of the reasons we think that calcium release happens from the SR in heart failure, despite the lower load, is that you still have, at certain stages at least, a retained beta-adrenergic responsiveness, where beta-adrenergic response stimulates the phospholamine phosphorylation and SR calcium pump. So maybe regionally and heterotypically or heterogeneously in the heart, you have activation of load that is sufficient to trigger spontaneous calcium release as calcium sparks or waves. And that release then is much more likely to cause a depolarization that is large enough to trigger an action potential in the setting of heart failure. So let me come back to the arrhythmogenic portion here. And so the unloading of the SR is contributed by these three characters here, but these two can also directly contribute to the propensity for arrhythmias. As I've said here, the exchanger is what carries the current that's driving the depolarization, and the leak may be what also during diastole is enhanced and triggering calcium release. So either the load can be triggering it by going up transiently, or even under normal conditions, a greater leak of calcium from the SR can start the ball rolling. So the question then is, how do you measure leak? So the notion that leak is important in heart failure came really from Andy Mark's group, based entirely on single channel recordings in black lipid bilayers of non-physiological composition. So we decided that if leak was important, we really ought to be able to measure it in an intact cell. And the idea there was, if leak is important in limiting the load, if we abruptly block the leak, the load should go up. And if we prevent transsarcolemmal calcium fluxes, as the load goes up, the cytosolic calcium should go down. So we block the leak abruptly with tetracaine. Here's a kind of experiment that Tom Shannon did when he was developing this protocol. So you stop stimulation, measure the calcium transients here, apply tetracaine or not. With tetracaine, diastolic calcium goes down. You do this quick sort of switching here to allow you to apply caffeine and dump the SR content. The peaks are about the same, which means that the total amount of calcium in the cell was the same. We've just shifted calcium from the cytosol into the SR. So it's out of the cytosol, this drop here, and that means the SR content was bigger. So the amplitude of this tetracaine-induced shift in calcium from cytosol to SR is indicative of how much leak there was to begin with. To block the leak, you shift the calcium. So as a function of the SR calcium content before you block the leak, we found that, in fact, at low loads of SR, adding tetracaine didn't matter. The load didn't go up. So leak is probably quite low when the SR content is low, but then it goes up steeply as the SR content goes up. And at this point here, you see this curve starts to get rather steep, and it's reflected in both the change in SR calcium load and as well as the diastolic calcium decline. And in heart failure, what we see is that in any given SR calcium content, this curve is shifted upward, there's a bigger leak, and the curve again reaches this kind of steep phase at a lower SR calcium content, which in fact may limit the SR load. So it's clear then that leak, and we could be more quantitative about exactly what this leak means in terms of a rate constant of efflux, etc., but the bottom line is still the same. At any given SR load, there's more leak in heart failure. So there is a component of SR leak that we see in the rabbit heart failure model I've described here, and consistent with the notion that leak contributes to unloading calcium from the SR, and so all three of these players be contributing to that, as I mentioned at the beginning. But how does this increase in SR leak occur in heart failure? Well, there's a popular story that Andy Mark's lab has propagated, and they have an elegant series of experiments to substantiate the story. But what I'll tell you today is I don't think it's the whole story. And that is that Pka-dependent hyperphosphorylation of the ranitidine receptor by the Pka, which is a member of a macromolecular complex that is attached to the ranitidine receptor, that phosphorylation kicks off FK binding protein here, FK506 binding protein, and that event increases the leak of calcium from the SR. So again, the data from that was simply was purely based on bilayer measurements at the time. We said, well, we can measure leak in the cells and see whether Pka in fact does stimulate the leak of calcium from the SR, and that might be what's wrong in heart failure in terms of the SR calcium leak that could both unload the SR and contribute to VAD. So the way we did that was the following. So the problem with Pka effects on the ranitidine receptor is Pka has multiple targets, and I've indicated three that are known and principally related to this EC coupling area here. One, of course, is it stimulates the calcium current. It phosphorylates phospholamine to stimulate the pump, and I've just shown you how much calcium is in the SR makes a big difference in how much leak there is, so we'd have to control those things. And so the way we've done that is in a two-step process. One is we permeabilize the cell to allow us to control the cyto-KMP and directly activate Pka in the cell, and also to control the calcium environment in the cell so that we can control and measure it. That also kicks or knocks the L-type calcium channel out of function because now there's no voltage gradient across the membrane, nor any calcium gradient for the channel to work with. So that takes care of that and allows us a modicum of control here, but retaining a relatively physiologic environment. And then to get rid of the potential pKa effect on phospholamban, we use mice that either have no phospholamban at all, so it can't be phosphorylated, or only have a mutant phospholamban that lacks the phosphorylation sites that are regulatory. So this phospholamban, in this case, the double mutant, would be a constant inhibitor. And in the knockout case, it would be gone so the pump can go at its maximal turnover rate. And then as another measure of leak, we measure calcium sparks. So that represents the cluster of ryanodine receptor openings, and we think that most of the leak is via sparks. But I guess I would posit it to you also that those measurements that I showed you before are a more comprehensive measure of leak than are calcium sparks. Nevertheless, we think that the majority of the leak does occur by these ryanodine receptor openings that we can detect as sparks. So what do we see? Well, so this is a line scan, and I'm sure you've seen these before, along the length of a myocyte. And then if you watch as a function of time, you see these spots, bright spots of calcium release, or calcium spark, I'm going to call them. And if you put on cyclic AMP, the spark frequency clearly goes up. So it must be that you phosphorylate the ryanodine receptor and it goes up. But here we have the wild type mouse where there's also phospholamban. So what happens in the knockout mouse, the spark frequency is a little bit higher to begin with, kind of comparable to this, and you add cyclic AMP and nothing happens. No increase in sparks at all. So we also did the experiment at a little higher calcium concentration to make sure we weren't seeing a ceiling effect here or a maximal spark frequency. We knew that wasn't the case. But if we go up to a little higher calcium, and that's why the shade of blue is brighter here or lighter, you see we have a spark frequency in the wild type cells here that's kind of comparable to what we have in all three of these panels. But we add cyclic AMP and it goes up even more. And it can keep going up and up until you have these big waves and macro sparks. But we do the same protocol with this sort of dominant negative or constant inhibitory phospholamban that can't be phosphorylated, and the spark frequency is kind of like that, and it doesn't change with cyclic AMP at all. Here's the pooled data from experiments like that. No phospholamban, no change in sparks. In the wild type, a robust increase in spark frequency of 200%. In the dominant negative or double mutant phospholamban, no effect on spark frequency, and the wild types a robust increase. So we reasoned that really phospholamban was the important thing in dictating whether this leak or calcium spark increase occurred. And indeed, we suggest then that it's the increase in SR load by stimulation of the pump, and this effect of luminal calcium somehow regulating and stimulating these release events, that's the culprit. And indeed, when we measure the SR content by caffeine-induced calcium release, we find that in the wild type there's a significant increase in the calcium content of the SR. In the knockout, there's not, and the same for the data here. So it looks like the phospholamban-dependent stimulation of the pump is the crucial step in increasing the calcium spark frequency, and that the ryanodine receptor phosphorylation, which occurs, and we verified that in these experiments, is not the culprit. Moreover, if we go back to intact myocytes and kind of just don't stimulate them and see what happens to calcium sparks, if you stimulate a wild-type myocyte with ISO, you still stimulate the pump, SR content goes up, cytosolic calcium goes down, and what happens is you increase spark frequency. In the knockout, you put ISO on, nothing happens to spark frequency. So we concluded from these experiments that pKa does not appear to alter ryanodine receptor leak as measured by calcium sparks in mouse ventricular myocytes. And these are normal mouse myocytes, not heart failure myocytes. So you could explain some differences between them in a lot of different ways. Bottom line is we haven't been able to see these effects. We also went back quite recently and redid these experiments with permeabilized cells and exogenous catalytic subunit of pKa, just in case there was some limitation in the activation of pKa here, even though we saw it robustly in the wild-type cells. And with exogenous pKa, same result. In the wild-type, an increase in calcium spark frequency. In the knockouts, no effect. So we've still been unable to see stimulation of sparks with Pka activation. Well, what about Chemokinase? I told you I would talk about Chemokinase, and now it's about time. So we do the same kind of experiments with Chemokinase, and I'll describe it to you here. Here's a resting calcium spark. This is in the phospholamban knockout. We've also done it in wild type. But this is kind of the crucial experiment. So you see some sparks, and what we do here is we pre-activated Chemokinase. So Chemokinase, without going into too much detail on the Chemokinase molecule itself, it has an autophosphorylation site, and once that site is autophosphorylated, the kinase becomes active and independent of calcium at a little bit lower level than normal, but it's still quite active, say 50 to 80% active. So we pre-activate by elevating calcium and calmodulin with some Chemokinase, and then we get it pre-activated and then dilute it back down to the calcium concentration that we have here. You can see the blue shades are the same. So we've matched the calcium, and it's exposed to this Chem plus Chemokinase solution, even though the calcium outside is the same. And so what happens over time, we put okadaic acid outside here to whatever gets phosphorylated stays phosphorylated, because we block the dephosphorylation by this phosphatase inhibitor, at least by PP1, PP2A, and certain other phosphatases. And so what you see is there's some increase in sparks, and we wash that out, and there's still an increase in sparks. So the okadaic acid has presumably left whatever's phosphorylated, and we know the ryanodine receptor is phosphorylated here, And what we see is that overall the spark frequency is increased by this cocktail, and it's further increased upon washout. And what I'll show you in a couple of moments is the reason this green bar is lower than this one, we think, is because calmodulin itself is inhibitory. And so the calmodulin that's in this cocktail and is later washed out actually depresses the calcium spark frequency. We also know that at the point out here where we're measuring this enhanced spark frequency by about a factor of 2, that the SR calcium content is not reduced or increased. So at a comparable SR content, there's an increase in calcium sparks dictated by Chemokinase, and that effect is independent of the load. Furthermore, if we include the peptide inhibitor of Chemokinase in here, we find that we actually do not see any enhancement, in fact a slight reduction on average of the calcium spark frequency. So there might have been even some basal level of phosphorylation by Chemokinase. So that actually encouraged us to say, well, okay, we can do it with an exogenous Chemokinase that we pre-activate. What about the Chemokinase that, as I'll show you later, is already bound to the ryanodine receptor? Can we do it with endogenous Chemokinase? And this is a little more challenging experiment because cells don't like to be in conditions where the Chemokinase is highly activated, but we cooked up this condition that seemed to work quite nicely. So same thing, a phospholamban knockout myocyte that has some sparks, not too common. And then we raise the calcium in the yellow region here up to 500 nanomolar and include about a micromolar of calmodulin in that. And that's just to ensure that there's plenty of calcium and calmodulin to activate the Chemokinase that's already sitting there. And then for 1 minute later, we wash that solution out and return to this solution here, and we see that there's a dramatic increase in calcium spark activity. And if we do the same thing, but now throughout this protocol, put a Chemokinase inhibitor peptide, AAP, in, we completely abolish it. So here's the calcium spark frequency immediately after the Chemokinase activation in the endogenous case. And if we include this AIP peptide in there, it completely prevents that increase in calcium spark frequency. And again, the SR content tends to be a bit lower, as you might expect if you had enhanced leak, but it wasn't significant in this case here. So it looks like Chemokinase is having a substantial activating effect on SR calcium release at rest, diastolic calcium concentrations. And in fact, even if we look more detailed at the calcium sparks, what we can see is that in this histogram here that's the fraction of events, so it's normalized to the number of sparks that were given. In the control case, there are almost no sparks at all that are longer than about 70 milliseconds in full width half max. They're all over before then. It's less than 1% cumulatively up here. Whereas in Chemokinase, you see quite a few of these longer events. so that the average, or the mean open from 50% is shifted several milliseconds longer. And indeed, there's quite a few longer events here than we see in control. So it looks like these events are more frequent and they're longer, and thus may contribute to the ryanodine receptor leak that could in fact lead to depolarization that we talked about earlier. So this could be a mechanism by which the ryanodine receptor gets activated in heart failure, and I will get into that in a few moments. Let me first show you the data I promised about when we do those experiments with just adding exogenous calmodulin alone. What we find is that in a concentration-dependent manner, adding calmodulin decreases calcium spark frequency. And in fact, there's a Ki of about 100 nanomolar, which matches the binding affinity of calmodulin to the ryanodine receptor, and bilayer data shows that calmodulin inhibits the ryanodine receptor with about this kind of Ki, and the spark duration magnitude are not much changed. So what about CAM kinase effects on the ryanodine receptor during EC coupling itself? That is, this is all diastolic resting calcium efflux. Well, a few years back, we did a series of experiments. Lee in my lab did these experiments. And what she did was to match the SR calcium load after conditioning pulses that activated the calcium in a way where we would expect CAM kinase to be activated to some tonic level. So she did those experiments with and without the inhibitor of CAM kinase KN93, and she was very careful to make sure at the time she was comparing the calcium transients during the twitch, the SR content was the same. So as measured by caffeine-induced contractions, integrated sodium-calcium exchange currents, peak sodium-calcium exchange currents, or calcium transient amplitudes, all four measures of SR content were the same before, or in the absence and presence of KN93 in the same cells, paired measurements. And what we saw was, for the same calcium current and the same SR load, if we block CAM kinase, we see a dramatic reduction in the fractional release, or the calcium transient, in this case contraction, same data for calcium transient. So endogenous CAM kinase and its activation during regular beating is probably important in maintaining this fractional release or efficacy of EC coupling in the normal situation. So CAM kinase then, and endogenous CAM kinase, seems to activate the ryanodine receptor to some sensitized level during normal EC coupling. So again, you could think that maybe what goes on when it's abnormally activated is it's oversensitizing and increasing leak during diastole, or if cells are quiescent, this deactivation of the CAM kinase system may further reduce the calcium transients that we actually typically see at very low frequencies. So we've also done the same kind of thing with Pka, because, well, it's another kinase that we know should alter the calcium transients. And in fact, it's well known that Pka activation by isoproterenol, in this case, activates bigger calcium transients, greatly stimulates the calcium current in these simultaneous recordings that Ken Ginsberg did. And that's true even in the mouse that has this phospholamine double mutant. So even though the SR pump can't be stimulated by this ISO, the increased current and increased SR calcium load gives you a bigger calcium transient. But for this experiment, we have the same problem as before. That is, Pka has multiple targets in this EC coupling context. And in this case, what Ken did was, rather than try and control specifically this and this or get them out of the picture, he did an exhaustive series of experiments where he varied SR load over a wide dynamic range, he varied the calcium current over a wide dynamic range, and then grouped the data by, okay, here's the cells that have the same SR content plus or minus ISO and the same calcium current. And the long shot of those results is that the SR content, when matched for low, medium, and high loads, plus and minus ISO, and we always made sure that if anything, the ISO group was a little bit higher so as not to bias the results in the wrong direction. The SR calcium release for any given calcium current interval, or peak current, was basically the same. So isoproterenol did not at all affect the fraction of calcium released during a voltage clamp pulse. And that surprised us, and it surprised Andy Marks too. But so the enhanced SR calcium release with Pka activation in terms of quantity is due to the increase in calcium current, which we know occurs normally when you activate the sympathetic system, and also the increase in SR content, but not necessarily to the ranitidine receptor phosphorylation per se. So we thought, well, let's look at the data a little more carefully. There's got to be something that pKa does here. And indeed there is. So if we look at the maximum rate of release of calcium, or the time to peak rate of release, that's accelerated by ISO. But remember, I told you the total amount released was the same. So what ISO seems to do, or Pka activation, is it speeds up the rate of release, but it still shuts off, let's say, at the same SR load. So the shutoff of release is dictated, and is also sped up. You get faster release, faster shutoff of release with ISO. And in a certain sense, if you think about what the sympathetic nervous system is doing to the heart, it makes kind of sense. It's speeding up the calcium release, but it doesn't appear to be affecting the amount if you control the trigger and the load, which is different than what I showed you for CAM-kinase. So PKA is definitely doing something to the release process, but it seems to be in a more dynamic sense. And this actually matches some nice results from Hector Valdivia a number of years back on biolayers, showing that ISO stimulated the peak of the calcium channel open probability, but not the steady state open probability. So in our hands, I'll summarize the results we have for PKA and CAM-kinase on these three key targets in the coupling. The effects of PKA that we see are, of course, dramatic on calcium current, dramatic at stimulating the SR pump, but rather modest on altering ranitidine receptor function. And data which I haven't shown you so much, we do see effects of CAM-kinase and calcium current facilitation and inactivation kinetics. CAM-kinase also phosphorylates phospholamban, but in our hands, the range over which that's controlled is kind of a more moderate effect, but exerts big effects on ranitidine receptor gating. So they both have the same three targets. They both probably phosphorylate different sites on those three targets, but the impact of those results seems to be rather different.
So John, you didn't touch on FKBP 12.6 or stable 2. I wonder why.
Well, so both in our hands and Andy Mark's hands, the CAM-kinase effect on the ranitidine receptor. So he sees also activation by CAM-kinase, but he doesn't see it dissociating FKBP. And I'll try and come back to that when I show.
I'm just curious to know what your view of this is.
Yeah, and we can talk about that some more, maybe in this summary part too.
Over dinner.
Over dinner, yeah. But I'll be happy to comment on the sort of the big picture issues about that. So looking more at CAM-kinase, there are some results several years back by a German group that reported in human heart failure there was upregulation of CAM-kinase expression in heart failure. And together with Joan Brown and Tong Zhang at UC San Diego, we looked at aortic banding in mice and looked at the expression levels of CAM-kinase and found that CAM-kinase was upregulated rather early in the time course after banding. And sort of to our surprise, the predominant form that was upregulated was the nuclear isoform, the CAM-kinase delta C. These are the two main isoforms in ventricular myocytes, not the only, but main ones. And this one has a nuclear localization signal, and the CAM-kinase delta C is usually the lower band when they're together. So it looked like even in rather short term, rather than chronic human heart failure, CAM-kinase is upregulated. In co-immunoprecipitation studies, we showed that CAM-kinase also comes in with this same big macromolecular complex with the ranitidine receptor, the same one that Andy Mark's group described nicely that includes PKA, PP1, and phosphatase 2A, and a couple of other proteins as well. And so it clearly is part of that same macromolecular complex, and several other groups have reproduced this result in the last couple of years. So that seems clear. And so in Jones' lab, they made mice that were transgenic mice overexpressing CAM-kinase delta C. And I won't go through a lot of details in the model, but just to say that these mice have a profound heart failure phenotype. In 12 weeks, about half of them are dead. And they have big hearts. They have the usual heart failure phenotype in terms of gene expression. They have a dramatic increase in ranitidine receptor phosphorylation, calcium sparks, and a reduction in SR calcium load. I'll show you a little bit of this data as I'll go on. So here's some of the contraction calcium transient data in these transgenic mice. So the wild type calcium transients, transgenics, are much depressed. You can see that here. The caffeine calcium transients are even further depressed. They're really small compared to the wild type, especially if you integrate the responses. And the fractional release is increased. That's this over this. So not surprisingly, that's increased. But that would be consistent with what I showed you before about direct activation of ChemKinase in carefully controlled voltage clamp experiments, that it's sensitizing the release. So there's an increased fractional release in this transgenic model and a reduction in load. So if I tell you there's a huge reduction in SR calcium load, what would you expect with sparks? You'd expect there to be very low sparks because they were so sensitive to load. And I will tell you that diastolic calcium in these myocytes is also lower than normal. Yet we see a dramatic increase in calcium spark frequency. They're a little lower in amplitude on the average, and we think that's because the load is down. But they're more frequent, and they're a bit longer in duration. Moreover, even at an earlier stage, before these animals have developed substantial heart failure or phenotype, the calcium spark frequency is up. It's up even more, in this case threefold in terms of frequency, and it's completely prevented by the ChemKinase inhibitor. So this is not an adaptive change in expression pattern. It's probably the acute phosphorylation, which we in fact measured on the ranitidine receptor here, at least at the ranitidine receptor. So that's consistent with the ChemKinase effects that I showed you before. Now, transgenic models have lots of inherent problems in terms of how things change over time and adjustments that the animals make to the altered gene expression. So we also did these acutely in 24-hour adenoviral overexpression of ChemKinase delta C to a similar level of activation as we saw in the transgenic mouse and heart failure case, which I'll tell you about in a bit, in rabbit myocytes. And the results are pretty similar. That is, twitches, calcium transients are down, not quite significantly. The SR content is down, this time significantly, but again, the fractional is increased. And to jump ahead a little, you see this caffeine, the SR content isn't down quite as much here as I showed you before here. And we think that the difference is probably because, in addition to the leak here, which may have been due to ChemKinase, there's also an upregulation of exchanger, downregulation of circuit, which we demonstrated in these. And those factors, as I pointed out at the very beginning, may all contribute to the unloading of the SR. So these guys have three hits on SR content, as it were, and that makes the SR load especially bad in these transgenics. The adenoviral cells don't have altered circuit or exchanger function, but they do also have an increased spark frequency at the same SR calcium. So let me come back to the heart failure case. And as was described in a previous study in human, in the rabbit heart failure model that we studied, we also see an increase in the expression of ChemKinase delta. In this case, we now know it's the delta isoform, and in fact, delta C especially, the cytosolic isoform. And that's true whether we co-IP the ranitidine receptor or just measure global myocyte ChemKinase expression. It's up by about a factor of 2. It's more heavily phosphorylated, so that's up also, and there's a reduction in calmodulin. And all three of these things these two things in particular here, would be consistent with the notion that there should be enhanced calcium leak from the SR as we measured experimentally. We measured ranitidine receptor phosphorylation using Andy Mark's antibodies for the 2815 ChemKinase site, and you can see that's dramatically increased in heart failure. And also the 2809 site, which he claims is Pka specific, and other groups have argued is also a ChemKinase site. At any rate, the phosphorylation is also increased at that site. And we've shown that in other ways too by back phosphorylation and P32 incorporation. So the ranitidine receptor is more phosphorylated, there's more ChemKinase there, it's more activated, and what about the leak? Well, so if this is all true and correct, and here's the same data I showed you before, in heart failure there's more leak at any given calcium load, can we simply block the leak by inhibiting either ChemKinase with KN93 or Pka with H89? and restore this curve over to this control curve where the leak is back down to the normal levels. So experiments that Tom Shannon and Jerry Curran did address that in the same paper that came out recently in Circ Research. And the idea is that if we look at any given SR calcium load, and again, we're going to use this strategy that we've got a bunch of data, and we're going to group it by where the load in heart failure and control was the same on the average, what was the amount of leak or tetracaine-induced shift in the calcium. So what you can see is that we've matched under three conditions, heart failure myocytes with CAM-kinase or pKa block. The loads are all about the same. And what happened to the leak under those conditions? Well, the leak was suppressed significantly with CAM-kinase blockade, but not with pKa blockade. We could look at this data another way, like going across this way and say, okay, well, what about for any given level of leak, what's the load, the SR load we can reach? Kind of getting at the idea of what's the maximum SR load one can get in these conditions. And what you can see is, again, with the matched leak going across horizontally here, you got a much bigger SR load when you block the CAM-kinase, consistent with the notion that CAM-kinase is inducing a calcium leak, and that PK is not really culprit here in the heart failure situation that we're studying. So, well, maybe we've cured heart failure by blocking CAM-kinase. Well, no. We have increased the SR calcium. So now these are field-stimulated calcium transients. So this is not like when we're controlling everything. We're just stimulating at a constant rate. And what happens is, indeed, the caffeine-induced calcium store goes up after KN93. You can see that here. And that's dramatic. But if you look at the twitch calcium transients, well, yeah, it's significantly higher, but not very much. So what we think is really happening here is, indeed, you have blocked the leak. But by blocking CAM-kinase, you've also desensitized DC coupling. And those effects almost cancel each other out. And that would be completely consistent with the notion David Eisner has proposed and shown very nice data for. Because there's a lot of autoregulation in this system, such that if you sensitize the ryanodine receptor, by any means, by sensitizing it to a calcium trigger, you may get a bigger release and lower load, but you also enhance the fractional release, and you end up with calcium transients that aren't very different. And I think that's what's happening here. And I think that's also why, in the heart failure case, despite the fact that SR calcium was low, we got a normal fractional release. Because the load itself would have depressed the fractional release. The CAM-kinase phosphorylation that we measured may have enhanced it somewhat. So we ended up with an unaltered fractional release. And we have that same kind of data in human heart failure. So it looks then like we've got these mechanisms that can unload the SR and contribute to the systolic dysfunction, and at least the enhanced exchanger function and SR calcium leak may contribute to arrhythmogenesis and trigger DADs in heart failure. And that the story may not be quite as simple as the one that Dr. Mark's group focuses on with pKa regulation. There may also be a significant role for CAM-kinase in modulating these processes. So far I focused on CAM-kinase and regulation in heart failure and arrhythmias, although I didn't talk so much about actual arrhythmias, more about the fundamental mechanisms by which CAM and CAM-kinase regulate ryanodine receptor. A little bit I've implied about how CAM-kinase regulates the SR calcium pump. I didn't talk about how CAM-kinase modulates calcium current and how the altered calcium current properties, the facilitation and slowed inactivation, may be involved in EAD initiation. Mark Anderson's group has focused studies on that. I've also talked about how CAM-kinase can contribute to the heart failure phenotype. But interestingly, I think the heart failure phenotype parts that it contributes are not systolic dysfunction. It unloads the SR, but it also sensitizes the ryanodine receptor. So the reason the systolic dysfunction occurs is probably by the SERCA and sodium calcium exchange functions, not so much by the CAM-kinase effect on the RYR. Having said that, I think it is true that the leakiness of the RYR can be datogenic. That is, it can mean that you get calcium release, and that increases the likelihood of DADs occurring, whether they're relevant or not. Okay, but I'd like to back this up down into it. Oh, wait. CAM-kinase kinase also has sodium channels, and that was to remind me to tell you about the data that I'm not going to talk about in the talk. So I'm going to back that out.
It came up in the Keystone meeting.
Pardon? Yeah.
It came up.
But I thought
There was some
would.
data from what's her
Yeah.
name, Isabelle Duchenne.
Pardon?
Isabelle Duchenne and Gordon Tomaselli, where they had some.
Right, but Lars Meyer and I have some data specifically with the same adenoviral transfer of CAM kinase that modulates the sodium channels in a way that's very analogous to that sodium channel mutation, the INS 1795D, that creates both the Brugada and LPT kind of syndrome. Anyway, so let's go away from this picture for a moment, because I want to talk about, in the final few minutes here, about nuclear CAM kinase and how it may also be involved in calcium release from nucleus stores and transcriptional regulation. So last year we published a paper showing that in ventricular myocytes, the IP3 receptors that exist there are almost exclusively the type 2 IP3 receptor isoform, and that they are almost exclusively in the nuclear envelope. In addition, they have a very high affinity for CAM kinase, and in fact, they'll find each other in free solution if you express them separately and mix them. They find each other and co-IP very beautifully. So this affinity is quite strong. And CAM has also been reported previously to interact directly with IP3 receptors. So this looks like maybe another little signaling complex here with another release channel. And in our minds, this really was stimulating because IP3 receptors have been known to be in ventricular myocytes for a long time, and they have no known function there. That is, we can't see any impact on systolic function when we activate or inhibit IP3 receptors in ventricular myocytes. So maybe their role has something more to do with a signaling complex here. And then Eric Olson's lab had this beautiful story about how histone deacetylases that can serve to repress transcription by MEF2 transcriptional factors upon phosphorylation, and CAM kinase is an HDAC 4 or 5 kinase. Upon phosphorylation, they're exported from the nucleus, and this repression is relieved, and it de-represses transcriptional function, and you can get hypertrophic or heart failure gene expression as a consequence of that. So we backed this story up a little bit and said, well, maybe if CAM kinase is involved, and maybe the IP3 receptor is involved, and then we need where IP3 comes from, which is from G protein coupled receptors and their phospholipase C, etc., which produce these guys here. So maybe one of the ways that these hypertrophic signaling neurohumoral factors like phenylephrine or endothelin-1 work is by IP3 and triggering this cascade here. So it seemed like a low probability. There were way too many steps that had to work. But I'll tell you that it kind of works. So we started by getting GFP-tagged HDAC5 by adenovirus into ventricular myocytes, adult rabbit ventricular myocytes. And you can see that it's primarily localized in the nucleus here, as expected at the resting condition. And here's a blow-up of that same image. And then if we expose the cell to endothelin without any stimulation, you can see that that HDAC5 is coming out of the nucleus. Not entirely, but about 40% comes out in just 60 minutes. If we go for 8 hours, it all comes out. And in fact, if we look at the time course of HDAC nuclear export with endothelin versus control, it's quite striking. At the same time, there's no apparent elevation of calcium in the cytosol. The cells aren't stimulated. We can't find any hot spots of calcium anywhere in the cell with confocal microscopy. So it looks like at least endothelin can cause HDAC nuclear export. What about any of these other steps? Well, let's start with the question of whether this signaling cascade has to do with IP3 or DAG or PKC activation. So the first experiment then was to say, well, let's block PKC and see what happens if this pathway is important. Well, blocking PKC by BIS didn't do anything. And this concentration should really knock out all PKCs. So we still have the same nuclear export. However, if we add an IP3 receptor blocker to APB, which is not the greatest drug, but I'll show you we have other evidence for IP3 involvement, it completely blocked the endothelin-induced nuclear export. if we block the reuptake of calcium into the nuclear envelope, and remember the nuclear envelope is a double membrane-bound organelle. It has circupumps and it has Ip3 receptors, as I mentioned. So if we block the circupumps from pumping calcium into the nuclear envelope, no response. If we permeabilize the cells and add adenosine, a potent and selective Ip3 receptor agonist, we can recapitulate the entire response of HDAC nuclear export simply by activating these Ip3 receptors. So the Ip3 receptor is sufficient in activating to produce this HDAC nuclear export. And is it necessary? Well, if we knock out the Ip3 receptor, and we have these Ip3 receptor type 2 knockout mice that Ju Chen, our collaborator in San Diego, generated, it completely abolishes the response. And by the way, you can see here that in mouse wild-type mouse myocytes, the response is pretty much the same as we saw in the rat myocytes, which were the bulk of the experiments. So it looks like the Ip3 receptor is involved, and it's definitely calcium sensitive. Is calcium released from the nuclear envelope? Well, it turned out that we had developed a protocol to measure intra-SR free calcium using a low affinity calcium indicator, Flu5N, which has an affinity of KD of about 400 micromole. So it only lights up the regions of the cell where where calcium is very high, millimolar concentration, like inside the SR. And in our initial studies, the nuisance was it also lit up the nuclear envelope. We weren't interested in that then. Well, it turns out we are interested now. And so we focused on the nuclear envelope, and if we're careful about the exposure conditions to the laser and the confocal, in 30 minutes we get pretty reproducible results here. And if we apply adenosine, the Ip3 receptor agonist, you can see that the nuclear envelope is substantially dimmer. And so we do see evidence of calcium release from the nuclear envelope associated with the Ip3 sensitivity and its localization. So can Ip3 get to the nucleus? This has been a story in the Ip3 receptor field that Ip3 gets broken down, and so can it really get from the surface to the nucleus? And to do that, together with Greg Minery's lab, we generated an Ip3 sensor by tagging CFP and YFP by the Ip3 binding Ip3 receptor binding domain from the receptor type 1 and type 3 actually. And so we get FRET then. So if we excite the blue fluorophore here, if these are far apart, we don't see much fluorescence energy transfer between them, and you see emission at 485 and not much at 530. But if Ip3 binds and brings them closer together, which is the direction that we happen to see, didn't have to be that way, then the excitation here can transfer energy to the YFP, and we see an enhancement of the 530 fluorescence or an increase in the ratio of 530 over 485. So we can get a nice calibratable curve for the Ip3 concentration and the ratio of fluorescence when we do these kind of FRET experiments. And what we can see is in neonatal rat ventricular myocytes with the plasmid, endothelin produces a rapid and robust increase in the Ip3 concentration. More to the point, the adult ventricular myocytes, which required an adenovirus to get the sensor in there, you can see with endothelin, phenylephrine, or angiotensin, prompt rises in Ip3 concentration in the cell produced by these agonists. And we can even get an idea of what concentration range they might be occurring, because if we do a patch clamp pipette here with high Ip3 concentration, which saturates the indicator, we can find out what the maximal ratio we get is. So it's about 12.5% here. And if we try and dialyze out whatever endogenous Ip3 there might be by putting no Ip3 in the pipette, maybe we get a minimum that's a little lower than the average baseline for the cells. So the Ip3 produced is in a range where our sensor is working. Moreover, we also now have the ability to measure spatial and temporal changes in Ip3 concentration in the cell. So if we focus on the nucleus and only measure the signal from around this nucleus here versus the cytoplasm here, and we apply endothelin, you can see that, in fact, in the cytosol, the concentration of Ip3 rises faster and to a higher level. There's a little lag for it to get into the nucleus, and it doesn't get quite as high. So the amplitude of the nuclear signal is only 2/3 as high as the cytoplasmic signal, and it takes time. It's partly delayed. Anyway, the half time is delayed by about 30 seconds. So it makes sense that the Ip3 might be produced at the cytosol, and it has a little trouble diffusing into the nucleus. So the Ip3 story makes sense from that standpoint. Well, then, if this is a calcium-dependent process, why doesn't it get activated with every heartbeat? So in this experiment here, we have GFP that's the nucleus, and we can measure calcium transients. And you can see here are cytosolic calcium transients going along here. The ROD2 signal we use to measure the calcium is also in the nucleus. It's a little concentrated there, and you see nuclear calcium transients as well. But with this field stimulation by itself, nothing happens to HDAC. It doesn't need it. So it was calcium dependent, but somehow it's insulated from these global calcium transients that occur both in the cytosol and in the nucleus. Well, can we raise calcium enough to induce it independent of these Ip3 receptors? Well, we can if we permeabilize the cells. At resting calcium, the HDAC stays in the nucleus. If we raise the calcium in the cytoplasm for an hour, which of course is completely unphysiological, but calcium can be raised without killing the cells at that level for a while. And indeed, you can see the HDAC is coming out. Not quite as much as the endothelin is, Ip3 agonist, but we can make it come out. So it's clearly calcium dependent. And we think that the answer really is that the local signaling going on here is akin to the local signaling we think about with EC coupling. That is, we know with EC coupling that the calcium released from one cluster of ryanodine receptors normally doesn't activate a cluster that's down here, because the activating calcium has to be rather high. The same thing may occur here, so that the calcium around an Ip3 receptor, right where it's released, can bind to calmodulin, activate Chemokinase. They're all in one protein complex. So locally, around that protein complex, the local calcium may be tens of micromolar at least, whereas if we try and look in a confocal image for how much calcium is going on, we're looking at a volume that's 10,000 times bigger, and we could easily miss the signal. So that's an excuse for why I can't show you pretty calcium release signals around a single Ip3 receptor. The signals are probably just too small. You probably need a cluster, like we have for ryanodine receptors, that work as clusters, and their flux is much higher as well. So we think it's local. What about calmodulin and Chemokinase? We can block it with calmodulin inhibitors. We can also completely block it with sort of global serine 3D protein kinase inhibitors. But those aren't very specific. What about, and the HDAC actually has to be phosphorylated. If we use an HDAC5 that has a mutation so the serines are converted to alanines where the phosphorylation normally occurs, it doesn't get exported from the nucleus. It just sits there. However, if we block Chemokinase with K93, we half inhibit it. If we block PKD with this inhibitor here, we also half inhibit it. And if we block both PKD and Chemokinase, we can completely abolish it. So it looks like this nuclear export is at least partially Chemokinase dependent, but may also be partly regulated by protein kinase D, or as used to be called PKC-mu. It turns out that PKD is the kinase that Eric Olson's group found was the most important in neonatal myocytes at activating HDAC nuclear export, more important than Chemokinase. Another interesting aspect is PKD actually is much more like Chemokinase in terms of its substrate specificity and binding domains than it's like any PKC. So there may be more analogy here. The surprising thing to us was the PKD effect also seemed to require calcium in Ip3. And up until now, no one has reported PKDs as a calcium-activated enzyme. Although rumor I hear is that a group in San Diego has such data now. So this is just a summary slide, and I won't go through it. I only want to point out one thing. Here was the K93 data, which half-blocked the nuclear export. And we also have some nice data with transgenic mice that have the same Chemokinase inhibitory peptide targeted to the nucleus. So it's only in the nucleus. And we see the same extent of depression of the HDAC and nuclear export. So all this data is really consistent with that overall model that I showed you before. And I'll come back to it in a second. Maybe I'll skip over this. This just shows that Chemokinase does get autophosphorylated, and that in fact the MEF2 reporter genes, MEF2 reporter activity is increased. So we are actually activating protein expression with endothelin in a way that's blocked by dapsogardin. So each step in this process that I talked about here, from endothelin on down to the HDAC nuclear export and relief of transcriptional repression, seems to be consistent with this kind of story, with the exception that it may not be all CAM-kinase. There may also be a PKD component to that process. And also that the calcium signaling is all about this local region here, just like we talked about local signaling in this area up here. So hopefully in this kind of trip around the myocyte, I've given you some insights into how CAM-kinase may be involved both in EC coupling, arrhythmogenesis, and SR calcium leak, and also excitation transcription coupling that may be important in hypertrophic signaling in heart failure. So in closing, I just want to thank the people who did the work. These are the people from my lab who did most of the experiments that I talked about today, and other people at Loyola who were excellent colleagues and collaborators on certain parts of these projects, and a group of collaborators here, especially Steve Pogg was on all the heart failure studies in Rabbit, Joan Brown, and Lisa Cranius, John Did, and Eric Olson, and McKinsey for either supplying mice or reagents that are used for these studies in collaboration. So thanks very much. Happy to answer. Yeah.
I have a question with the data that you just showed with K93 being able to block the nuclear CAM-kinase to prevent the HDAC export. So if you treat one of your heart failure rabbits with K93, do you see any partial or full recovery of the phenotype?
We haven't done that yet, partly because we're hopeful that there will be better CAM-kinase inhibitors, because those are both very expensive experiments to do in terms of the amount of drug and sort of chronic treatment of the animals. And also, these agents are somewhat non-specific, yeah. So it'd be nice to do it in another way than just KN93.
Because I'm not sure if you would support this statement, but would you say that the nuclear, what's going on with the nuclear isoform, that's kind of more of a major player in expression of the heart failure phenotype?
Yes, I would say so. Probably that. Here I put CAM-kinase delta B, and we're not even sure that this is the way the IP3 receptor is spaced, but my gut and working hypothesis is that this CAM-kinase delta B is crucial in part, at least in certain ways, that neurohumoral signals get to the nucleus. I'm sure that, obviously there are other pathways, calcineurin pathways, and that got cut off here because there's probably another type of calcium signal, not the same as this, that's important in calcineurin and NFAT signal. So I think that what activates nuclear regulation in hypertrophic heart failure is very complicated, and this is one of probably many pathways. And there may be some other ones that also involve calcium and calmodulin. This is one where at least we think that the particular pieces have been worked out. I think conversely, and surprisingly, these CAM-kinase effects from the cytosolic turn out to probably be as precipitating in the initial heart failure phenotype by creating potentially this hemodynamic stress. That is, you overexpress CAM-kinase delta C in the cytosol, which may be partly a result of overexpression for a positive reason, like stimulating things, but it turns out to be negatively effective in terms of EC coupling. Yes?
So all calcium spots, are field stimulated, or some spontaneous?
Most of the ones I showed here today were impermeabilized cells. So they're quiescent cells where we buffered the calcium to somewhere between 10 and 100 nanomolar free calcium, and there's actually a significant amount of EGTA in those as well. So there's no stimulation in those cases. I can't think. Well, I showed some data with intact cells as well, but not stimulated. So we measure sparks not during stimulation, but after stimulation stopped.
Did you ever see calcium wave?
Sure.
in the workout mice,
Yes.
the transgenic mice.
Yes, in the chem kinase overexpression mice, we did, and they're harder to quantify. We did take the extra effort in those calcium sparks where we did permeabilize cells and applied exogenous or endogenous. I showed you how the average events were longer, but we also counted what we call macro sparks, which are kind of too big to be a single spark spatially, and also mini waves. The waves don't travel so far in that solution because we have EGTA there, but when you get a wave, that's a pretty robust, under those conditions, a wave is a very robust calcium release event, and we see substantial number of macro sparks and mini waves with chem kinase and none in its absence.
What regulates the expression of the different chem kinase?
That I don't know.
Anything known?
Yeah, not much is known about what are the regulators of chem kinase expression. That would be relevant for this particular pathway, because it could well be that these pathways are kind of reinforcing, and that this whole HDAC nuclear export system is not an initiator, but a reinforcer of a hypertrophy or heart failure phenotype, and we just don't know that yet.
On the chem inhibition of the ryanodine receptor, do you know if it, at the single channel kinetic level of the ryanodine receptor, do you know if it decreases the number of openings, or does it decrease the mean open time? Do you know
So
how?
Gerhard Meissner has done the most careful work on that on bilayers, and sees, I believe, that it doesn't, that it affects the open time. So with CAM, he gets a slight shortening of the typical open time of the.
That's from Gerhard's data?
Pardon?
From Gerhard Meissner's data.
Correct. And we were unable to detect that if the spark characteristics were not significantly different, but that could easily be masked by the sort of macroscopic microscopic behavior of sparks. Macromolecular microscopic.
Any other questions, comments? Well, if not, then thank you again for taking us on a